Looks good. Okay. All right, I think it is right at the top of the hour here, according to Verizon's United States Air Force Space Command atomic clock. It's 11 o'clock. So, we were talking about different sort of ways of understanding and knowing, right? And we ended on Thursday talking about the nomothetic deductive model. That is, starting with general theory and going to testable application. And kind of the last thought we had before closing things down on Thursday was, okay, this is great. This is the Sherlock Holmes approach, right? Elementary Dear Watson. And, you know, we get Benedict Cumberbatch and Robert Downey Jr. movies and stuff like that out of this. And that's all well and good, but there's a whole other way of doing stuff that is just as useful for different stages of the research life cycle. So let's talk a little bit about that research life cycle and kind of locate deductive versus inductive reasoning on that. So let's see, get this console all set here. There we go. So we have deductive reasoning, moving the cycle clockwise in our sort of cycle system. And then the inductive approach starts with data, not really a hypothesis. And it starts to get to, by the end of the inductive research cycle, forming a new grounded theory, because you're building it from the ground up instead of a top-down deductive approach of some big grand general theory and see what trickles down to your specific case that you're testing out in your experimental setup. Okay, so that's deductive reasoning, clockwise around the circle, and inductive is counterclockwise. And the sense of where to use these in kind of the life cycle of research is as follows. So with the life cycle research, we have kind of three stages. We have exploratory research, which I mentioned on Thursday, is what we generally tend to apply inductive research methods and the inductive research paradigm to exploratory research because it's some new phenomenon, maybe that we've just discovered. And we don't know much about it at all, so we don't have enough information to make a worthwhile hypothesis to try and test on it. We don't even know how the thing works yet. We have no idea. It's a totally new phenomenon, right? Think of the first day that humanity was aware of the possibility of things like black holes. So we're just trying to get any kind of sense or information we have about this. That's exploratory research, and so we start with data collection. And then from there, we'll eventually get to, you know, a theory of relativity that gets us to some kind of useful, quantitative, pretty detailed mechanical descriptions of black holes and singularities and so forth, event horizons and all of that. Schwarzschild radiuses, radii. But until we start even figuring out that black holes exist, we can't start putting together ideas like what is the radius around a black hole within which collapsing into the black hole as light, as matter, whatever, it's inevitable. You cannot get out with any conventional force in the universe. So induction versus deduction, pretty straightforward. But our topical life cycle is going to help us decide which of those major research paradigms to use. And so as researchers, we tend to gravitate towards one or the other, Some folks really like being out on sort of the bleeding edge of science where it's all sort of murky and indistinct, and we have, you know, from our previous research projects leading up to this, really conflicting information and insights and sort of really competing theories, right? There's no covering law or general theory about it. And some people really like that sort of cowboy Wild West sort of, we don't even know what we're going to find. Here we go, you know, hold my beer. Kind of an approach, but also very careful science in doing that, not just, you know, cavalier about it. Versus at the far end of the spectrum, where we know a lot more about a phenomenon. We call that explanatory research. We're doing very carefully controlled lab tests using positive controls, negative controls, controlling for placebo and nocebo effects. We're testing multiple different kinds of treatments in parallel and multiple different levels or intensities of treatment within each of those parallel treatments. Think about how we try to cure individual kinds of cancer or treat individual kinds of cancer, right? The medical trials are extremely detailed in their organization because they have to be, right? We are dealing with people's lives and survival hanging in the balance. So we want to know everything about that particular cancer that's under study. We want to have detailed molecular, biological, mechanistic models of when we get this extra functional group tagged onto this molecule that we're eating or using as a food preservative or dye or whatever, that functional group solubilizes that particular food molecule or preservative or synthetic molecule, whatever it is, across the cell membrane into the cell nucleus. That functional group, breaks off, just dynamites itself into the person's DNA and causes cancer or tumors. So carcinogenesis, teratogenesis, or mutagenicity. And we need to understand the phenomenon down to the, submolecular level to understand, okay, why are all of the people in this one village in Russia, why are they all only having girls as children? There's a specific, traceable reason. We need to figure out the mechanics of that, okay? That's a real-life series of studies.
Excuse me, but how is it possible to survive if you take up only one gender?
That's the concern, yes.
So,
that's the problem.
So question is, how are you getting girls if only girls?
So in the natural world, you may be familiar with a bunch of excreted hormone studies, water quality studies, and effluent discharge downstream of like industrial sites where hormone-like or hormone-suppressing chemicals make it into water. Famously in the United States here upstream of us on the Mississippi River. The people living in Minnesota and Wisconsin are taking things like birth control, which is hormone-based. And those hormones can, at very, very, very low concentration, at nano concentration levels, parts per billion, they can affect human physiology downstream. And for many, many years, water treatment plants just sucked in water and killed all the germs in it without necessarily doing what's called tertiary treatment to remove other chemicals that other folks upstream had put into the water. And so it was changing the expression of things in folks downstream.
So technically speaking, this is chemical castration.
A very, as we would say in English, a low-key form of not castration, but definitely altering in measurable ways the characteristics of the downstream population. Yeah, for sure. So since the 1950s, serum concentrations of testosterone in male humans has been slowly dropping because we have been drinking a whole lot of industrial chemicals before, in particular, the passage of the Clean Water and the Clean Air Act. And we were drinking stuff that was not real good for us. These days, right, we eat about a credit card of plastic a week. And part of the chemicals in those plastic nanoparticles are going to be endocrine disruptors, hormone hormone mimickers, and so forth.
So that kind of side effects of water is like permanent or temporary? So if you drinking clean water, eventually you're going back to normal testosterone levels or not?
So that's an open question. We're all running an experiment on all of us by default. It's exploratory research, very exciting. We don't know where it's going to go. But
yeah, really exciting and
yeah, it's all very exciting. So in the beginning, the inception of a sort of humanity-wide effort to study any particular phenomenon, whether it's a species in Florida or whatever it might be. First, we got to figure out what it is, right? So we're just collecting data on basic sort of introductory descriptors of like how many of this species exist, how long do they tend to live, and so on and so forth. What makes this species unique or distinguishable from other adjacent species, maybe in the same, genus or family, something like that? Are we sure this is genuinely a different species, or is it just a weird subpopulation that all have the same mutation, but they can interbreed with others of the larger set of species? We're just trying to feel around the thing with whatever sensory inputs that we have, whether they're sensors or senses. And exploratory research tends not to be published as much as descriptive or certainly explanatory research because it's a lot more nebulous. At the exploratory phase, typically you don't know what you don't know, right? So we have known knowns, things that we know, things that we know we don't know, known unknowns, and so forth through the Latin square of unknown unknowns. In exploratory research, the square for unknown unknowns is necessarily pretty big, right? We're just starting to grapple with this new thing. And then in the middle, we have descriptive research. So a lot of field environmental research is descriptive in nature. It can be up to quasi-experimental. Experimental research is explicitly and exclusively explanatory research because we're trying to break the mechanism to prove that we understand the mechanism. But descriptive research is necessarily as far as we can take certain kinds of research. So for example, a lot of astronomy research, they talk about running experiments on the Hubble or the James Webb Telescope or whatever. But it's really descriptive research. They're not able to experimentally control the stars that they're observing. The signals that we're observing from those stars were emitted, in some cases millions of years ago. And so we're just observing and describing what is observed. So where are those stars? What frequency, what's the spectrograph of essentially the colors of electromagnetic radiation that they're putting out? Radio frequency pulsars, X-rays from black hole gamma rays from like supernovae, stuff like that. But we can't run an experiment on those stars. They're distant. We can't touch them. We can't affect them as far as we know right now. So descriptive research tends to be the kind of research where there's something important that you need to know. And so you're doing the research to figure out how to describe what you need to know. A lot of human social research is descriptive research because we can't take everybody who voted in the 2016 election and run experiments on them in a controlled way and see how the election goes differently with more or less or different types of foreign misinformation efforts and that sort of thing. So this gets weird and interesting and exciting. And a lot of what we're going to be doing in forestry and wildlife and so forth is going to be descriptive or kind of this weird spot in between descriptive research called quasi-experimental research, where we are trying to run experiments on maybe paired watersheds, where we're controlling, this one's clear cut and this one isn't. Let's check the water quality draining out of each watershed. Or some of the original paired watershed studies. But we're not fully controlling everything that could be different about those two. We're picking two that are right next to each other, so they're experiencing the same climate. but one's going to be slightly larger than the other. They're going to have a slightly different mix of vegetative cover species, a slightly different mix of cyanobacteria, algae, things in the water column of the watershed draining out from those areas. And so that's quasi-experimental, right? We're running an experiment on two paired watersheds. One's the control, one's the treatment. But we don't have full laboratory sort of hermetically sealed airtight control over all of the variables present, which would be kind of the gold standard ideal. And so for explanatory research, we have a lot more control of almost everything, typically. So medical research is going to be controlled as much as it can. But in randomly assigning some patients to a control group or best current available treatment, not just like a sugar pill, for cancer patients or whatever, and other folks to some new experimental cancer drug, we're going to try to pair folks so that, person number one in the control group is very demographically similar to person number one in the experimental treatment group. Both female, both 18 to 35, both, insert race and ethnicity combination, both in the same general area and stuff like that, but they're not the same person, right? They have different genetic profiles. They're going to respond differently to whatever molecules you're trying to introduce to their bodies to control that cancer. So quasi-experimental stuff is going to be ultimately a lot of what most people think of as science and experiments. But if it's not fully controlled, it's not a fully controlled experiment, right? Tautologically. Yeah.
Technical question. So basically, what and who cannot be placed into exploratory research also? Because what and who, it's like a person and not person, so maybe we can apply that question from descriptive to exploratory, or not?
Yeah, you can. So the very first time anybody looks at a phenomenon, we're not even maybe to the point of being able to ask, okay, for this social phenomenon, the rise of neo-Nazi groups in America, so modern day fascist movements in the United States. First, the moment anybody first asks the questions, do we have, National Socialists in the United States at all? That's more of a presence or absence question. We have to get to the point of where are these groups tending to form? What's the timeline of formation? Who tends to self-select into those groups? And later on, we'll be able to understand enough of this descriptive stuff to start asking, how does somebody, in certain socioeconomic situation or cultural or media inputs, how does somebody wake up one morning and think, you know what, I think I'm going to be a jackbooted neo-Nazi and express my dissatisfaction with the state of things. But we need to know a lot. We need to be able to describe in a lot of detail the when, where, and who. And even up to that point, sometimes we just have to start asking the question, all right, should we be even asking about neo-Nazi presence in the United States? Not yet anyway. Should we even be asking that research question? Is that a thing? That's more of just a what question. So we can kind of split hairs a little bit. We can sort this very finely. But yeah, we could conceptually, have the who, the membership in the exploratory research. But sometimes we don't even know to ask that yet. We have to figure out, is this a valid phenomenon, or am I just imagining a blip or a statistical outlier that doesn't signify anything meaningful? It's just a bump in the data set. It's just noise where I think it might be signal. So yeah, that's a good question at a conceptual level. Yeah.
If I understand correctly, We cannot identify main, how to say it, layer or how to say it, without circumstances of phenomenon. We cannot identify who is causing that phenomenon. Yes, am I right? Or
Correct. Yeah.
if we wanted to understand who is doing something, we need to understand circumstances of phenomenon. Yeah.
Yeah. Yeah, and the circumstances would typically be when, where, who, what's the size of it, right? How many people is part of the who question, and so forth. So the where can be a literal geographic cluster or location. It can be a conceptual location. Where on the political spectrum do these people come from, or what have you? So imagine, you're scuba diving as a world-traveling grad student, and you see a really weird-looking fish, and you snap a picture of it with your water-resistant iPhone 15 Pro, and you upload that to not just Google image search, but things like any of the wildlife citizen science networks where anybody can upload data and get things identified and contribute to sort of global data sets. And nothing can identify this fish. Like there's 0 hits on Google that are clearly correct. Nobody among the ichthyologists that you share this photo around to who are familiar with that region where you went scuba diving, none of them say, I've ever seen anything that looks even remotely like this. The fish scaling pattern and the overall physiognomy, the shape of it, they're like nothing I've ever seen. You might have a new species, right? But it's ambiguous. It's not clear yet. We don't have all the information. We just have a photo. And we're trying to figure out, is this a new species at all? And if we get enough information, maybe some other photos, or maybe a 100% lack of any other photos, and it is genuinely a new discovery in terms of a species, well then that tips us over into the very interesting questions of, is this an anadromous fish? Where is its habitat? How many individuals are we looking at? What are the characteristics of it? What are the descriptors? So exploratory research is for people who are very comfortable with and even excited by ambiguity. It might just be that nobody's ever gotten a picture of this fish, but we really know what it is. It's just the only other description of it was in 1874 in some textbook that nobody has reprinted since then, and it's discovered, but we don't really know anything about it. Versus descriptive research, you're getting back on that dive, you're dragging nets, you're looking for more of those fish to start categorizing what's actually going on. Yeah.
You gave very interesting example about this fish, because now the question is, in this case, how to distinguish real facts with pseudo facts? Because, for example, there is like zoology and cryptozoology. So, technically speaking, how to distinguish pseudo facts from actual facts?
That's
Because
a
the
great question.
cryptozoologist will be, oh, I told you like 1,000 years ago, you didn't listen to me. So how to deal with that situation?
Luckily, we've already discussed that there are plenty of alternatives to research. This is just a very, very short and incomplete list. But for example, if we have people who are absolutely convinced that Bigfoot exists,
Excellent.
Yeah.
Excellent example.
Based on grainy movie film from the 1950s or 60s, whenever that was, we would call that premature closure.
Mm-hmm.
Somebody saying, I am absolutely sure that this thing exists. I've seen the video, right? That video is empirical data, but a premature closure person will say that's empirical evidence, right? Data and evidence are not quite the same thing. So evidence explains things under a certain theory. data are just data. They're just numbers, they're just observations, they're just film frames, they're just whatever they are, and so forth. So the difference between pseudoscience and actual science research are going to be the presence or absence of these terrible techniques, right? So if we say, you know, I've got this picture of this as yet unidentified fish in this particular area, so I bet this fish probably exists all around the entire sea, the Caribbean, the South China Sea, whatever, Mediterranean, whatever it is. That's overgeneralization, right? We've got one picture of one fish.
Yeah.
We have no idea where its habitat is. We haven't done the actual research of finding those descriptors of that species. What is its range? What is its habitat range? And so forth. Okay? So there are very clear markers between science and not science, which are very, very frustrating for us scientists, or we scientists to, us scientists to encounter in the wild. There was a guy calling into a financial advice like YouTube channel, which is interesting on its own, but he was making some poor financial decisions, and he had discerned, because he was a professional astrologer, he wasn't making any money, but a professional astrologer, he had divined that he was not making any money because like Jupiter was retrograde into something else, like, and you know, the viewers of that YouTube video, or really of that web show, you know, we're all rolling our eyes so hard, like it's back into our heads, we're looking backwards. Like, of course, you know, we don't think that the relative positions of heavenly bodies has anything to do with anything, including personal choices, good or bad, wise or unwise, of individual humans here on Earth, right? We just don't think that, because we don't have any evidence for that, right? But that person feels like he does. And so that might be maybe not any of these things, possibly the halo effect, right? So there is a sense of mysterium tremendum et fascinans, as the Romans or later theologians using Latin would say. The fascinating and terrifying mystery of reality. Stars are cool. The solar eclipse coming up in April is really cool. So astral bodies moving in relation to each other, that's pretty cool. I wonder if that really cool thing affects my life. Halo effect, a spillover effect of, I think astral bodies are cool. Let me interpret my life in relation to something that some of the rest of us don't think has any relevance to it. Okay, so does that kind of answer your question? Like pseudoscience versus science? Right. Yeah, so, you know, it's easy for us to sort of throw up our hands and laugh at somebody who's into astrology if we are thinking about, you know, serious social theories or the cognitive psychology of personal money management, decision making, and stuff like that. We can do carefully controlled lab research on that stuff, and we'll talk about, you know, some weird Nazi experiments and weird experiments about Nazis that try to do stuff like that in an actual scientific way, but that led to some harm a little bit later in the class. And we get to take a look at things that we kind of wish were pseudoscience, but were honest efforts at the time. Just we hadn't got the state of the art on ethics up to snuff there. Okay, so anyway, back to our discussion. So exploratory research is, again, it's less commonly published. In the language of publications, you'll see these two terms. It's a pilot study. It's a preliminary study. They may go so far as to explicitly identify it as this is an exploratory study. We're still working on this, but this is what we found. This is new, it's exciting, or it's really scary. You know, we need to get the word out right away, and we'll keep on investigating this. As we get more of that descriptive information, we can move into more detailed research with it. Okay, so a pilot study is a small study. Pilot study might be, or a pilot plant in chemistry or chemical engineering might be the size of this room or this building, and you're working out the kinks on producing whatever chemical it is, figuring out temperatures and flow rates and mixing techniques and reagents and catalysts. And then once you got that pretty well working, the chemical engineers come back in, took a look at everything that the pilot plant is set up to do, or the pilot research is set up to do, and you scale it up times 10,000, times a million. And then you have not just a chemical factory, but a chemical complex, right? So SIU has a bunch of pilot research facilities at the Dunn-Richmond Center, for example. They're doing like coal liquefaction, extracting coal hydrocarbons into water and doing all kinds of cool energy and fertilizer research with it. And they think they can do some pretty amazing stuff with that. So they're currently looking for funding partners to take that. It's a room about twice the size of this room, and got all sorts of steel machinery and research equipment in there, and turn it into, you know, not a 1 gallon per hour kind of a thing and a whole bunch of manual interventions, somebody twisting all the knobs to make each batch work. They'll get it automated and, you know, 100,000 gallons an hour kind of thing. Or, you know, millions eventually, right? So all of our oil refineries started out as pilot plants a century ago, and so forth and so on. So when you see a preliminary study mentioned, that's not bad research. It's not pseudoscience. It is just at the beginning of the process. There's nothing wrong with that. Typically, you might have in the course of your research, maybe once you're a PhD, might have 2 or 3 pilot projects going on continuously the rest of your career. And maybe 1 out of 3 of those turns up something interesting or useful or controversial or surprising. And so you take that pilot study and you scale it up into a full-scale study, and you figure out, you move into the descriptive and later the explanatory stages to get some good work done on the pilot study that worked. If a pilot study doesn't turn up interesting data, this is very important. This is very important. If you don't write any other notes today, write this down. If a pilot study doesn't give you the result that you were looking for, where the statistical tests come up negative, there's no difference from the null hypothesis in the pilot study, that doesn't mean the research failed. It doesn't mean you're a bad scientist. You've done the correct thing by ruling something out, and you still need to get the word out about failed or negative result studies. Because if you don't, somebody else is going to have to do the same thing. Because if you're wondering it, somebody else is too, somewhere in the world, right? So we waste a tremendous amount of the most highest and best potential of humankind by duplicating research because the word doesn't get out.
So, short comment. Shortly speaking, results are results, nevertheless the expectation.
Yeah, the results are whatever they are.
Yeah.
Yeah. According to the logical positivist paradigm. Okay, so moving on. So first we want to get to, we want to use this explore, describe, explain, research life cycle thing. Really, practically speaking, this is about getting to your research question, right? So my current PhD student, Rebecca, the solar eclipse research question that she has lined up for April here, we were able to find 1 other study anywhere ever that asked anything even close to her particular research question. And the total stack of literature that speaks to what she's looking at is about this thick printed out. It's about 20 studies total, right? So this is exploratory research. Now, we are using survey questions from previous well-validated, well-explored, fully hammered out research, statistically validated, stuff like that, and published many, many times. But it's never been used for this particular research question. And so in her case, it's exploratory research, and we don't know what we're going to find. And that's okay. That's really exciting. When you are doing something that you can't find much basis for one way or the other, because nobody else has ever looked at that in that way, it almost doesn't matter what you find. Because if nobody else has any data that speak to it, nobody else can say that you're wrong, right? You've collected data and you can go somewhere with it. And maybe we don't find any difference among the different people groups that Rebecca's question is being applied to, but maybe we do. Who knows? The only way is to find out. Either way, even if we don't find any interesting contrasts between Eclipse viewers and folks who don't express that they're interested in it at all, we still got to get the word out about that. Like, this is a really unique event. Why isn't anybody interested in this? Why doesn't it have any strong effect on people? Whereas in previous cultures, all the way back to the beginning of record keeping, solar eclipses matter a lot. Like a lot, right? In every major religious tradition, they are there. In most every major world surviving mythology, eclipses are there. So we think that we should find something. And if we don't, that's even more interesting than finding the obvious result of, yeah, people are excited about the eclipse. Okay, next research question, you know? So again, a lot of interesting and important research happens with the, I didn't expect that. Not the huge groundbreaking thing. Usually that, I didn't expect that is the entry point to the groundbreaking, stunning research, okay? So look for things that surprise you. Look for things that you don't expect. Look for things, and be careful about this, that you disagree with and you sort out while you're analyzing your data, right? If you have 1 million samples and one of them is totally unlike all the others, maybe the batteries were low in the detector that day. Maybe not. Maybe it really was an alien. Yeah.
So basically, how to identify here various, how to say the major and minor issue. Like your result is falling out from your set, and you're outing that out. But how to distinguish various that something really out or shit happens? How to distinguish this? Because it can be something minor or something major.
You're not going to like my answer. So what you're asking is, at least in quantitative research, called outlier analysis, or outlier discrimination. So you are basically looking at say, a point cloud of data on, in this case, a simple two-dimensional plot, and all of your observations seem to be correlated in X and Y, explained in this case by an approximately linear relationship, right? So if we put a least squares straight regression line through this data, it looks something like that. Have a negative y-intercept and some slope off of the x-axis. When you have an outlier, the outlier might look like this, way over here. And so that's a really big outlier. Like that's, for those of you who've taken your grad stats classes, that's like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 15 standard deviations away. That's like a 1 in a trillion chance for that to be a random error, if the machine recorded this data correctly. So you go to the machine and you check the diagnostic logs. What was the battery level on this at that time? Or we recorded this data point at the exact moment I tripped and fell on the solar panel powering this thing. There was probably a bit of a power surge as I cracked that monocrystalline silicon, you know, powering this thing. Or you've got several outliers, and they're all behaving the same way. We had a solar flare that day, and it screwed up everybody's stuff. It even knocked out some satellites that day, right? Global communications disruption, and we happened to find it as well. So this happened to Kodak in the 1950s. I think it was the 1950s, yeah. The United States government was detonating atomic test weapons and not telling anybody about it. And Kodak, in those days, was shipping film across the country in train cars, open, unsealed boxcars, and they were packing the 35 millimeter like cellulose film in, I think it was corn husks. Before we had like styrofoam packing peanuts, we just used clean farm silage to pack things for long distance shipping. And Kodak was, the chemists at Eastman Kodak were ripping their hair out trying to figure out why is a whole bunch of our film showing up partially exposed, just these little speckles totally overexposed in the film, but only on certain train shipments. We know it's not light getting through because that would expose the whole section of film, and they're in closed boxes, and so on and so forth. And it was actually Eastman Kodak who figured out, wait a minute, there are radionuclides on some of this film, and we're pretty sure that these are not naturally occurring because they're only on very specific batches of train freight. And as it turns out, like the government was detonating nuclear weapons or test weapons. The fallout would hit these trains and the train conductors and everybody else living nearby before we understood what fallout was really about. And some careful ruling out of it's not a real thing. It's not a manufacturing error of the film because all the other film made at the same time in the same way in the same facility, but going a different place, that's all fine, and so forth. It's definitely something that's happening to the film in transit, right? So you're ruling out things that could explain, is it just random error, right? So you're likely to get some random error that moves the point this far. Not likely to have random error that moves several points this far, okay? But there's no specific single cutoff number that says, if it's here, it's too far away from the central tendency, that regression line. That must be a real thing. We got to figure out what that is. The government's detonating secret nuclear weapons. Very exciting and scary. This does happen 1 in 10 million times, something like that.
Okay.
As a result of random chance, random error.
So, like exactly on this example, how would you establish that the distance from dot is far away enough away? You understand what I wanted to say?
You said it yourself. You establish that 5 is too far away, 5 standard deviations away.
So,
that's a choice, right?
Okay, so for one dot is 5 deviations away, and another dot is 20 deviations away.
Uh-huh.
And how? So basically, how to identify necessary amount of deviations to confirm that that outliner has some kind of a meaningful impact.
So the way that you do that is, as the researcher, you're looking at the size of your data set. Let's say you're doing a pilot study, and your entire sample size, the total members in that point cloud, is 100 individuals. Or for social pilot studies, 9 individuals. The standard deviation in that case, which divides essentially the error or the offset by the number of samples in that set, it's a small number in the denominator, and so your standard deviation is going to be huge. Now, in explanatory research, where you have 50 billion members in this, it's a way smaller standard deviation size. So that means you're more likely to have something meaningful going on if you have one or a cluster of points away from the rest of the group. So there's a whole family of advanced statistical analysis that kind of sidesteps the issue of, or is used to examine the issue of outliers and so forth. And so, you know, we'll talk a little bit more about different statistical families of techniques later in the semester. Don't want to get too sidetracked off of it, but exploratory factor analysis and confirmatory factor analysis, EFA, CFA, they're used to figure out, is this the one group, and there's nothing systematic. There can always be random error, but if there's nothing systematic in the variables and the data that you collected that explains this. So cluster analysis figures out, okay, we've got this lobe of data. This is either a separate lobe of data or it's error. Cluster analysis is great for that. Factor analysis says, okay, you measured 3 input variables, and this is the result that you got when you plot them out. 2 of those 3 variables don't speak at all to this. For those variables, this looks like error. For the 3rd input variable, it explains this and this, and the other 2 don't at all. So we're going to combine those other 2 factors into what's called a latent variable, one that you didn't measure, but that the 2 variables you did measure, they're kind of indirectly getting at the real thing. Right? So the latent factor is the real factor that your measured factors are kind of speaking to, but not actually directly measuring. And saying, okay, your other variable, your other input factor, that looks like it maps pretty directly onto this and this. We're explaining both. So there are statistical techniques. K-means clustering analysis, confirmatory and exploratory factor analysis to help tease out when you get weird stuff like that. But fundamentally, before you roll out the very heavy tools of EFA, CFA, and cluster analysis, you as the human researcher are making the decision to roll those out. You're saying, that's a typo, and I'm just going to delete that point because I can't know what that point actually was. I clearly wrote it down wrong in my lab notebook, and it's lost to time, whatever. I wrote the digits in the wrong order, and I can't be sure what the actual order of the digits is. It's called a transposition error in digits. Or you as the researcher saying, no, I'm very confident that I didn't write this down wrong. I'm very confident we didn't have a power surge that day. I'm very confident I measured something real in those points. And the fact that they're 15 standard deviations away from the central tendency, I think that means something. I think I'm onto something here. So let's roll out some of these other statistical techniques and figure it out. Or if we're not even that far, if we have a small data set and statistical techniques are not the right move, in exploratory research commonly, I need to collect a ton more data. Now that I think there's something out there, let me do version 2 of this study, or generation 2 of this study, to explicitly, let me look over here now. I was looking here with this range of conditions that I tested, this time frame that I looked at, this area that I looked in. Let me look over here as well. There might be a whole other population that I just happened to catch a few of. And like the real big cluster is actually here, and I just blundered into, this is the small weird side population that we just didn't know much about either way, but until we started exploring it and finding weird behaviors. Okay. Last question. We're almost done.
Okay, yeah. So you showed an example of two axes.
Uh-huh.
But what if, for example, you have situation with three axes, like three criteria, x, y, z.
Uh-huh.
And on the two axes, you have that kind of situation, but on the axis z, another factor, it's kind of probably makes sense.
Yep.
So how to deal with that kind of situation when you have three factors?
Then that's generation 2 of the research, right? The first effort, the exploratory effort, was I think these two variables matter. So I'm going to plot things according to those two. As it turns out, I've got something. This is a two-dimensional slice of an n-dimensional, infinite dimensions space. So if we add a third axis on, it may be that the point cloud is actually a curve that intersects this plane twice, once with this population, once with this subpopulation. And posting that third thing, now we can see the real curve in space, or the coordinate space. Okay. We got to end, we're out of time. But we will continue with this. You're asking a really good question. This is getting at the heart of science. That's weird. This doesn't match our previous model. I might be onto something here. Okay.
Thank you.
Cool. I'm excited. This is cool stuff. This is what gets scientists out of bed in the morning. That and copious amounts of caffeine.